Is not just an important trial for breast cancer, although it definitely is that, but it is a model for innovation in drug development.
This is a real paradigm shift.
Quite literally, life-saving and game-changing.
It's a process actually more than it is a trial. It will set a standard, I think, and a very high bar for trials and processes to come.
Over the last 25 years, there has been an explosion of new and vitally important anti-cancer drugs. The development of these promising new therapeutic agents is generally based on preclinical and clinical research. In many cases, this research has become prohibitively expensive, and only a relatively few investigational drugs have reached the market and successfully improved clinical outcomes in the treatment of patients with cancer and hematological malignancies. In the development of new therapies, the traditional clinical trial process of determining which drugs will ultimately benefit patients is long and expensive. Over the last few decades, scientists have tried to change and improve the way clinical trials are conducted. Their purpose? Improving the process and improving the way novel pharmaceutical drugs are being developed. But it still takes many years and huge investments to successfully bring a new drug to market.
Currently, it can cost as much as $1 billion and take as long as 15 years to bring a new drug online.
But despite the strong efforts, there is still a huge unmet medical need.
The unfortunate reality is that for those women with the most aggressive cancers, they still face the stark reality that there's a significant chance that they will die of their disease.
While there are many novel drugs being developed to help improve the outcome and improve survival, resources are limited. Optimal use of resources requires better understanding of cancer biology, the identification of novel therapeutic targets, and the ability to address inefficiencies in the cancer clinical trial system. This may especially be so in how we treat women with metastatic, high-risk breast cancer.
We believe that our best hope now for these high-risk breast cancer patients is to develop targeted drugs to actually identify the changes in the genome that are responsible for the dysregulation of the genome that leads to cancer. We believe that if we can establish the biomarkers that indicate what is really going on in terms of the disease, that we can actually signal back to our drug developers what the biology is, then we can take years off this process and reduce the cost of developing new drugs for breast cancer.
Based on the current limitations in how we conduct clinical trials, scientists of the Biomarkers Consortium at the Foundation for the National Institutes of Health are changing the way new anti-cancer drugs are being developed.
We're seeing a change in attitudes in a number of ways, not the least of which, of course, is that industry is collaborating here, many pharmaceutical companies coming together for the same purpose. And this is a sea change. This is 10 years ago, this wouldn't have happened.
The Foundation's unique and groundbreaking re-engineered approach to clinical trials is the I-SPY clinical trial. This trial represents an unprecedented and streamlined method in developing new anti-cancer drugs. Using breast cancer treatment as a model, the I-SPY trials are designed to significantly reduce the overall cost, time, and number of patients required to bring innovative anti-cancer agents to the right patient at the right time, and to do this faster. The I-SPY trial program integrates and links Phase 1, Phase 2, and eventually Phase 3 clinical oncology trials to build a pipeline of novel anti-cancer agents. As a result, the new trial program accelerates the process of identifying a subset of high-risk breast cancer patients that will directly benefit from these novel agents. Among the targeted drugs being investigated in the I-SPY 2 trial are antibody-drug conjugates, or ADCs. Antibody-drug conjugates are part of a new wave of targeted antibody-based products. They are novel, innovative agents at the cutting edge of oncology and hematology. During the annual meeting of the American Association of Cancer Research, held April 16th through the 20th, 2016, in New Orleans, we sat down with Dr. Angela D. Michel, Professor of Medicine and Epidemiology at the Perelman School of Medicine at the University of Pennsylvania. We asked Dr. D. Michel a number of questions about a specific part of the I-SPY 2 trial, in which researchers investigated a particular antibody-drug conjugate and how a combination of drugs, including antibody-drug conjugates, can bring a substantially greater proportion of patients to the primary endpoint of pathological complete response, or PCR, an outcome in which, following neoadjuvant therapy, residual invasive cancer is detected in the breast tissue and lymph nodes during surgery. And we asked her about the benefit of targeted therapy and how novel drugs like antibody-drug conjugates play a role.
I think this is really an exciting time for antibody-drug conjugates because we have learned so much biologically about tumors and the immune system, and the idea that we can target tumor cells both with antibodies that have specific and unique activities on extracellular receptors, but also use them as a drug delivery device for highly toxic chemotherapeutics that we couldn't have given systemically, but now we've got a way to actually use these very unique linker technologies to be able to home in on the tumor cells and deliver the drug that would have been too toxic to normal cells specifically to the target. That's a really exciting and potentially very powerful approach. And TDM1 is just one example of this, where we already had an antibody, trastuzumab, that we knew was active on its own against HER2-positive breast cancer, that we understood a lot about the biology of the HER2 receptor, HER2 dimerization, and then all of a sudden now to have the technology to bind ematansine, which had been tested as a chemotherapeutic on its own and was much too toxic for people to be able to tolerate, now to be able to link that to this effective antibody and deliver it specifically to HER2 overexpressing breast cancer cells is really novel and revolutionary. And you get not only the delivery system for the chemotherapeutic, but you've got also the activity of the antibody itself. So you've got that 1-2 punch that really provides a very powerful way of targeting the tumors. And in this world of precision medicine, where we're looking for things that are unique to tumors, that are not found on normal cells, this is a very powerful way of saying, once we've identified these antigens on tumor surfaces, and now we've got the ability to design antibodies to those, then being able to link additional payload to those antibodies really has tremendous potential that I think we've barely begun to tap into. So I think that this is a time where we are going to see more and more of these therapies developed, not only because they are so potent against the tumor cells, but because they spare normal cells. And in the world of solid tumor oncology, I think that over the last 20 years, certainly we've made many strides of developing chemotherapeutics, cytotoxic agents that are effective against tumor cells, but we've seen that those off-target effects on normal cells like the bone marrow, like the heart, like the lungs, really can create later problems for patients that can really be quite debilitating. So a woman who develops cardiomyopathy and congestive heart failure 10 years after her treatment with an anthracycline just has a new problem now, even though she may have potentially been cured of her breast cancer. So being able to link an anthracycline to an antibody and be able to spare the heart really means we can take a drug we've already tested and know is effective against the tumor, but now what we're doing is really making sure that we get it right to where it needs to go, and we don't have these collateral effects that really are so damaging to patients. And the same is true with other types of toxicities. Neurotoxicity, for example, which is a major problem in cancer therapeutics, particularly in breast cancer, where we use a lot of neurotoxic agents. Neuropathy is debilitating for patients. They can't wear shoes, they can't type, they can't cook, they can't do self-care, and these can be not just numbness or tingling, but actual pain syndromes. So then we are getting into the realm of using narcotics and other kind of pain-modifying agents. So you could see that this really just begins to snowball, and the patient is left very debilitated, even if we've cured the cancer. So we've got to think both about how we can advance cancer, effective cancer care, and at the same time minimize and reduce the number of long-term complications we're giving to patients. We have many cancer survivors. That's a great thing, and that has really been the result of all of the advances in cytotoxic therapy over the last 20 years. The next 20 years has to be about continuing to improve that therapy, but sparing patients the side effects, the long-term complications that really impact their quality of life. If they're going to live another 30, 40 years, just as we hope they will after a cancer diagnosis, we want that to be a high quality 30 or 40 years.
In an effort to make this possible, scientists have developed a novel trial approach called the I-SPY trial.
So the I-SPY program began about 15 years ago, and initially this was an observational study of women who were receiving neoadjuvant treatment for their breast cancer. So the idea here is that when a woman is diagnosed, particularly with a large, aggressive, node-positive tumor, we knew that we could give chemotherapy before surgery in an effort to try to shrink the tumor and make the surgery more effective. Because 20, 30 years ago, we thought of breast cancer as a surgical disease. And we thought that what we needed to do was eradicate all of the disease in the breast. And of course that's true, but why do women die of breast cancer? They die of breast cancer because of micrometastatic or dormant cells that leave that primary tumor and get into the periphery, get into the circulation, ultimately find their way to other sites. So metastatic breast cancer that's gone to the bones or to the organs, that's what's lethal. So the idea was that if we gave chemotherapy up front, not only would we shrink the tumor, but we would also get this effective therapy into the rest of the body to try to start to attack these micrometastatic cells before they could take up residence in another organ or in the bones. And so when we first started to use this neoadjuvant approach, the approach was to use standard chemotherapy. And it turned out that if you gave the exact same chemotherapy regimen before surgery as after surgery, that the patient outcomes would be just the same. So essentially what that was telling us was the chemotherapy is going to be effective for some fraction of patients, and it didn't matter whether you gave it before or after surgery to see that effect. So what else can we learn? Well, what we can learn by giving the chemotherapy first and seeing its effect on the tumor is how sensitive the tumor actually is to the chemotherapy. And as time went on and more studies were using the neoadjuvant approach, we started to see very clear evidence from many studies showing that women who had achieved what we call a pathologic complete response, no tumor in the breast, no tumor in the lymph nodes at the completion of this therapy, did very, very well. They had a much better prognosis than women who had residual tumor in the breast and lymph nodes. So now all of a sudden we have a surrogate for a long-term outcome. That meant we could start to think about using this for drug development. Because if we have a short-term surrogate, pathologic complete response, that can tell us pretty reliably whether a patient's going to relapse later on, then we can start testing drugs against that surrogate. And if they're successful against the surrogate, then it stands to reason that they would be successful in terms of improving survival and reducing recurrence risks. Now, unfortunately, it's not that simple. Nothing is. So one has to really understand the surrogate. And the early years of I-SPY were really about understanding how much of a change in the tumor, what degree of response would correlate ultimately with an improvement in long-term outcomes. How good a surrogate was it? And ultimately what we found, along with many others at the time, but one of the principal things that I-SPY found in the very early years was that the relationship between response in the tumor, pathologic complete response, and ultimately relapsing from the tumor was very specific to the kind of cancer, the kind of breast cancer that a woman had. So if it was a triple negative breast cancer, lacked estrogen, progesterone, HER2 receptors, then there was a very tight correlation between getting a pathologic complete response in the breast and not relapsing later on. The flip side of this was if the tumor was estrogen receptor rich, full of these estrogen receptors, one might not see very much response in the tumor at the time of surgery, and yet those patients would still go on to do very well. Why would that be? Well, that's because of the advent of adjuvant endocrine therapy. So even if the tumors weren't very sensitive to the chemotherapy, patients were getting 5 and now 10 years of an anti-estrogen after surgery that was helping them to avoid recurrence. So it was a very instructive time in the early 2000s where many groups, I-SPY, the German groups, many different groups were really trying to understand this relationship. And this all came together about 4 years ago when the FDA actually did a meta-analysis, taking all of the studies that had looked at pathologic complete response, or PCR, as an endpoint, as a surrogate endpoint, and really looking at how good a surrogate it was. And they did that because they wanted, the FDA wanted, to be able to use this early surrogate to give provisional approval to drugs that looked very promising. And if we had to wait 5 or 10 years to see if patients were going to relapse, you could imagine that it would be very slow in terms of getting these drugs to patients. So having a reliable early surrogate is something the FDA would very much like to be able to use. So we were involved in those discussions and really trying to codify how you would use PCR as an intermediate endpoint for provisional or accelerated approval of drugs. And so now moving into the therapeutic aspect of I-SPY, what we are doing is now adding drugs or substituting drugs for the existing drugs to see if we can improve those PCR rates. Can we get a greater proportion of women to the point of having a PCR at the time of surgery? Because if we can do that, it would stand to reason that we would save a greater proportion of women from having their breast cancer recur.
A better understanding of cancer biology and how to use chemotherapeutic agents before surgery, also called neoadjuvant chemotherapy, led to patients achieving pathological complete response, or PCR. This will change the way physicians treat women with breast cancer. But how can the trial guarantee that women with breast cancer have a pathological complete response at the time of surgery?
So we moved into the I-SPY 2 trial in 2010, and it was specifically designed as a randomized phase 2 trial to help us find drugs that would significantly increase the PCR rate above what we would find in standard care, with the idea that if we could increase the PCR rate with these drugs, that ultimately they would be beneficial for patients in terms of long-term outcomes. And so the trial is unusual in the sense that first, it's a standing platform trial. That means we set it up in 2010, and it's been running continuously ever since. And it's designed to enable drugs that are being evaluated to enter the trial, go through a period of testing, have us understand whether they work or not, and then they leave the trial. And so 11 different drugs have come in and out of the trial over that period of time, and 5 of them have been found to be effective. And now that essentially tells us that these are drugs that we ought to then take forward into a confirmational phase 3 trial. So it's really a drug screening program, if you think of it that way, to take promising drugs, test them in this very early setting before women become metastatic, which also brings up an important issue. Drug development in the past had been done primarily by testing drugs on patients who are at the end stages of disease, who had stage 4 heavily pretreated disease. And if a drug were active in those types of patients, then we would think about moving it earlier in the disease trajectory. The problem with that approach is that, as we've learned now, the biology of the disease changes over time, and the molecular and genetic characteristics of the tumor at the time it's diagnosed in the breast are very different from the tumor that now becomes metastatic in other parts of the body. So drugs that work in the metastatic setting might not work in an early disease setting. And more importantly, drugs that could work in the early disease setting and actually have the potential to cure patients might not work in the metastatic setting. So using the metastatic setting as a screen for active drugs really was not a very effective strategy, particularly as we start to get into more targeted therapies. So understanding the biology of the disease and the advent of these more targeted therapies, as well as then a clearer understanding of this PCR surrogate, all coalesced to enable us to really start this trial that had the goal of adding new drugs to the existing regimens.
The I-SPY 2 trial allows drugs to enter and leave the clinical study quickly and seamlessly, allowing researchers to learn faster about better performing anti-cancer drugs and, depending on a tumor's molecular characteristics, treat participants more effectively with targeted drugs added to existing regimens. Prior studies have shown that a combination of drugs including T-DM1 was safe and effective against advanced metastatic HER2-positive breast cancer. In the I-SPY 2 trial, the investigators tested whether this combination would also be effective if given earlier in the course of treatment, before surgery.
So one of the unique things about the I-SPY 2 trial is that we can test multiple drugs simultaneously. We can test drugs that are specific to HER2-positive tumors, drugs that are specific to HER2-negative tumors. We can test the drugs within a whole variety of different breast cancer subsets and see where they work best. So in the HER2 space, there's a desire to really be able to bring a greater proportion of patients to this endpoint of pathologic complete response. We do pretty well with chemotherapy, usually a taxane-based regimen, and Herceptin. So it stood to reason that once the T-DM1 molecule came along, that would be very appealing because it's targeting HER2, but also delivering a very potent microtubule disruptor, similar to what we do with taxanes, but now delivering it right to the tumor. And so the goal of this arm of the I-SPY 2 trial was to test T-DM1 in combination with another antibody, pertuzumab. And these two have very synergistic mechanisms. While the T-DM1 is binding to the Herceptin binding site on the extracellular HER2 and absorbing the M-tansine, pertuzumab is binding to a completely different site on the HER2, and it's blocking heterodimerization. So they're really working quite synergistically. Moreover, they have very different side effect profiles, so it was very easy to combine these drugs together. So what we did was we compared the combination of T-DM1 and pertuzumab to the standard part of neoadjuvant therapy that would consist of paclitaxel, trastuzumab, and all of those patients went on to get the rest of the standard regimen, which was adriamycin and Cytoxan, and then they went to surgery. And the goal was to see whether the T-DM1 pertuzumab could increase the pathologic complete response rate over the standard paclitaxel trastuzumab. And in fact, what we found was that it did do that in a very significant way. Now, one thing I should tell you about the I-SPY 2 trial is that it's an adaptively randomized trial. What does that mean? It means that we use Bayesian probability statistics to adjust the randomization probabilities for patients coming into the trial. So we are constantly using what we learn about patients who've been treated by the drugs in real time to then determine what drugs patients should get the next patients who are coming into the trial. So what happened was that over time, during the time that T-DM1 pertuzumab was being evaluated in our trial, it was found to be superior to the other regimen. And we use a relatively unique endpoint in addition to pathologic complete response. We say that the degree of improvement in pathologic complete response that we see with the T-DM1 pertuzumab over standard paclitaxel trastuzumab needs to be sufficiently large to predict that doing that large confirmational phase 3 trial, we would still see this effect. So this is built on probabilities, a lot of longitudinal modeling, and really a very sophisticated way of saying not just, well, what are the absolute numbers? What is the PCR rate of the new combination to the old combination? But in fact, is the new combination good enough that we would expect it to be effective later in ultimately preventing patients from developing metastatic disease? So we call that graduation in the trial. And essentially it means that there's an at least 85% probability that TDM1 pertuzumab would be successful in a subsequent phase 3 trial. So that's the very nature of the screening aspect of I-SPY 2, a phase 2 trial that identifies drugs to take forward into phase 3. And if the combination does well enough, we know with high confidence that it will succeed in that confirmational phase 3 trial. And so we did find that it was superior and in fact increased the estimated pathologic complete response rates over the standard therapy by about 30 percentage points, and that it had anywhere from a 90 to 94% chance of being successful in a subsequent phase 3 trial. And this is the kind of thing that the FDA wants to see, because this enables them to say, here's a drug that we ought to support in a phase 3 registration trial. So many drugs that go into randomized trials ultimately fail. So having the information that you have a high likelihood of success coming out of a relatively small number of patients, we had only 83 patients in our trial, really is going to help direct both investigators and the FDA to the most active drugs and to really put our limited resources of clinical trials into trials that are going to be successful. And this is what happened with the drug pertuzumab. So pertuzumab was found also to really dramatically increase the pathologic complete response rate over standard paclitaxel and Herceptin. Pertuzumab, when added to those two drugs, actually had a very large impact, and that was a large enough impact that convinced the FDA to give accelerated approval to pertuzumab. Now, of course, the confirmatory phase 3 trial is ongoing. That's called the Affinity trial, but it's a great example of the way that a neoadjuvant trial can give the FDA sufficient confidence in the benefits of a drug that they will give it accelerated approval, make it available to patients who need it right now while we're waiting for the confirmational trial. And so what we found in our trial is that TDM1 pertuzumab really has that capability, and it is in fact being tested in several large randomized phase 3 trials. But the other unintended benefit, I think, is the fact that we were able to give up the paclitaxel. And so now we have a fully targeted regimen, pertuzumab and TDM1, that does not have the side effects on normal cells that using paclitaxel would have. And so what we found in our trial is that the patients who got TDM1 and pertuzumab had much less of the type of bothersome side effects like hair loss and neuropathy that really matter to patients. Patients felt very good while they were on this treatment. They weren't losing their hair, their hands weren't getting numb, they were feeling good, and they had very few other untoward side effects. And so what I think is really the bottom line here, the big news, is that we found a drug combination that can both increase the likelihood of patients being cured of their disease and at the same time give them fewer short-term and long-term side effects than the standard therapy. And I think that is ultimately what we all want to be able to do for patients. It's not just improve their outcomes, but also make sure we're preserving their quality of life. It's not enough to simply cure them of the disease if they're left with debilitating long-term effects. And that is really the goal, is now to build on this. So TDM1 can now become the new backbone. And because it is so non-toxic as an antibody-drug conjugate, we now can start to add other things to it in a way that we might not have with standard cytotoxic therapies, where you had to worry about all of these conflicting side effects. And so we are really enthusiastic about taking this backbone of TDM1 forward and starting now to build the house on top of that till we get to the point where every patient with HER2-positive disease will hit a PCR. Every patient can have that opportunity for long-term success.
The unique approach of the I-SPY 2 trial is that it uses adaptive randomization and a statistical approach called the Bayesian probability of superiority versus control to assign patients to the standard therapeutic approach or the investigational drugs. This phase of the I-SPY 2 trial demonstrates that by replacing older, non-targeted therapies with more effective, less toxic, new and targeted therapies, such as antibody-drug conjugates, it is possible to both improve outcomes and decrease side effects, creating a real survival advantage for women with breast cancer.
45 years ago, cancer was almost invariably a death sentence. It was feared. It was something that was felt to be the major threat in our time. We lacked the tools to be able to fight it. Over the impending 40 years, we then developed a whole set of tools and cytotoxic therapies that started to now make it possible for patients to survive cancer, and that was great. But the new technologies, particularly antibody-drug conjugates, now make it possible to take those effective therapies and deliver them in a way that won't have the toxic side effects that patients have been left with. In addition, we've learned so much more about the biology of tumors that we now have a cornucopia of antigens against which we can develop antibodies, continue to advance the linking technology, to link it to new payloads. And I think that that is going to provide us the opportunity to start to combine these antibody-drug conjugates in ways that can really attack tumors in multiple directions, again, with very little long-term and short-term toxicity to patients. So I'm very enthusiastic about the direction things are going. Certainly we've made a lot of progress, but we've got a long way to go, both in terms of eradicating the disease, certainly now that we understand more about the biology, targeting it in a very precise way, being able to then look at an individual patient's tumor and understand what are the targets for that woman. Not just what targets are out there, what drugs could we try, but in that very personalized sense, what is her tumor really all about, and how can we pick from these very targeted therapies that group that's going to eradicate that tumor. And that's where I see things going in the future, to really take this really powerful technology and begin to really apply it in a very personalized way to patients.
Personalized medicine offers a promise to deliver optimal patient outcomes by integrating clinical and molecular patient data to understand the biological basis of cancer. It guides the selection of the most appropriate targeted therapies based on distinctive patient characteristics and unique molecular features of a malignancy. Following this approach, the I-SPY 2 trial combines personalized medicine with a novel investigational trial design to identify women at high risk of early breast cancer recurrence. The trial is a new model in the development of novel anti-cancer drugs, a life-saving, game-changing approach, a paradigm shift, creating a real survival advantage for women with breast cancer. Next, in Changing Strategies in the War on Cancer, Part 4, the role of patients and patient advocacy.